All right, welcome back. Starting our discussion of the synapse or synapse. The synapse is the junction that mediates the information transfer from one neuron to the other to another neuron, or from a neuron to an effector cell. We've seen the synapse when we discussed the muscle contraction, if you remember. As the neuromuscular junction, we were releasing a neurotransmitter called the acetylcholine that binds to the receptor on the muscle cell membrane or the sarcolemma, and this causes the opening of the sodium channels, creating another electrical signal on the muscle cell. So if we are looking at the connection between two neurons, we consider the one that precedes the synapse. It's the presynaptic neuron that will be conducting the signal towards the synapse. And we consider the ones that comes after the synapse. It's the postsynaptic neuron that will be receiving the signal from the presynaptic neuron and transmitting the signal away from the synapse. So the connection between the neurons might be between an axon of a neuron, presynaptic neuron, and the cell body of the postsynaptic neuron, because this is an axosomatic. Soma means body. So the connection here between the axon and the cell body is what we call axosomatic. Axosomatic means connection between the axon terminals and the cell body of the postsynaptic neuron. Axodendritic, it's the connection between the axon terminals with the dendrites of the postsynaptic neuron. I might have a connection between two axons, but actually not the two axons terminals. The axon terminals of the presynaptic neuron are going to be connected with the axon hillock of the postsynaptic neuron. We call this synapse. It's the axoaxonic synapse. Axoaxonic, the connection here is between the axon terminals of the presynaptic neuron and the axon hillock of the postsynaptic neuron. Those synapses are going to be chemical synapses. What I mean by chemical synapse, it's the synapse in which the presynaptic neuron is going to be releasing a neurotransmitter from its axon terminals, and those neurotransmitters going to be binding to the receptor region in the postsynaptic neuron. So if I'm connecting an axon terminal to the cell body, so the receptive region of the postsynaptic neuron is going to be the cell body. If I'm connecting the axon to the dendrites, so the receptive region for the postsynaptic neuron is going to be the dendrites. This is where I've got the receptor for the neurotransmitters. The synaptic cleft is the fluid-filled space between the pre- and postsynaptic neurons. And this gap between the two nerve cells will be serving to prevent the direct electrical conduction between the neurons. So I don't want to have an electrical signal traveling from this nerve cell directly to this other nerve cell. So I have a tiny gap between the two nerve cells that are connected here. We call this tiny gap between the pre and post synaptic neurons. It's the synaptic cleft. So what happens here at the chemical synapse? I'm receiving an electrical signal, a depolarization signal, propagating along the axon. Remember, it will reach the terminal boutons or terminal lobes in the terminal ends of the axon. This electrical signal is going to be opening calcium channels, allowing calcium influx in the terminal end of the axon. This calcium influx is what's going to be stimulating the neurotransmitter vesicles to fuse with the cell membrane and to release the neurotransmitter in the synaptic cleft. Neurotransmitter, as you see on here, is going to be binding to its receptor on the receptor side of the postsynaptic neuron. Once it binds to its receptor, it will allow the opening of associated channels. See on here, so the receptor for the neurotransmitter is also going to be associated with an ion channel. So it allows the opening of the sodium channels and the opening of the potassium channels. So would I keep them open forever, creating an unlimited number of action potentials here in the postsynaptic neuron? No. What you're gonna do to this neurotransmitter, you either degrade it, you break it down, or you're gonna reuptake it back to the presynaptic neuron to terminate its action. Because if I release the neurotransmitter and it is kept attached to the receptor, what's gonna happen? It will create many more signals in the postsynaptic neuron, and it will have a prolonged action. I don't want to have this prolonged action, so what will I do? I will degrade the neurotransmitter, or I will diffuse it away from its receptor, or I will reuptake it back by the presynaptic nerve cell. How would I be terminating? Three different means of the termination of the action of the neurotransmitter: either break it down, degrade it by enzymes, reuptake it back by the axon terminal or the astrocytes, or those neurotransmitters gets diffused away from the synaptic cleft, so they won't be in direct contact with their receptors, and as a result, no effect gonna be resulting from them. Neurotransmitters, those are more than 50 different types that get released from the nerve cells, and they are classified according to their chemical structure and function. The first neurotransmitter and the most famous of them, it's the acetylcholine. If you remember, this acetylcholine is the neurotransmitters that we've seen back when we studied the muscular contraction, skeletal muscle contraction, that got released from the somatic nerve cell and caused the contraction of the skeletal muscle cell. It was located at the neuromuscular junction. Also, we're going to see the acetylcholine later on in the autonomic nervous system, as we discuss it in a later chapter. Acetylcholine gets deactivated and degraded by an enzyme called acetylcholine esterase. This is the main mechanism by which we're going to be terminating the action of the acetylcholine. It gets broken down by acetylcholine esterase. Another class of neurotransmitters, those are the biogenic amines. Those are like the catecholamines and indolamines. Catecholamines are like the dopamine, norepinephrine, and epinephrine. And indolamines, those are like the serotonin and histamine. Those are broadly distributed in the brain, and they play an important role in the emotional behaviors and the biological clock. Some amino acids act as neurotransmitters, like the gamma amino butyric acid or GABA, glycine, aspartate, and glutamate. Some are peptides, means short chains of amino acids. Those are like substance P, which is the mediator for the pain signals. Endorphins that will act as normal opiates. And the endorphins on here are going to be the ones responsible to reduce the pain perception. So if you are a totally healthy person and you don't perform any physical exertion at this point, are you actually in pain? So if I'm sitting in front of you, am I in pain right now? I am in pain, but I don't perceive it. So all the time, cells are going to be broken down. Your blood is rubbing against your blood vessels. You have fibrous tissues that will be broken down. So all the time we are in some kind of pain, but we don't actually perceive this pain because of the presence of the endorphins that get released in the brain. And this explains why people who are taking external opiates, once they stop taking those drugs, they will have exaggerated pain perception. So they will be in pain, although they don't have anything that should normally trigger any pain. Why? Because simply when a person takes external opiates, his brain stops producing the endorphins. And when he stops taking those external opiates, and no endorphins are going to be released in the brain, so he is going to be passing in a stage where I don't have external opiates and I'm not producing endorphins, so my perception of pain is going to be increased. Gut-brain peptides, those are signals that will be sent from the gastrointestinal tract to the brain and vice versa, and those are like the somatostatin and or somatostatin and cholecystokinin. Cholecystokinin, from its name, it enhances the contraction of the gallbladder. So chole means bile, and cysto means bladder, and kinetic means movement. So this is a kinin. Kinin means the protein that enhances the movement of the bile bladder. So it's the neurotransmitter that enhances the gallbladder contraction to squeeze out the bile that helps with the digestion of the fats. Novel messengers are like ATPs. We find out that ATP also might act as neurotransmitters in limited cases. Nitric oxide and carbon monoxide. Those are much less common compared to the previous ones that we've mentioned. How do we integrate those nerve cells together? We integrate the nerve cells in functional groups that will allow us to process the information and to conduct the signals that got processed to other destinations. So it might be integrating the nerve cells in a divergence pattern. It's a divergence in the same way. So I've got one input and I've got many outputs. I might be integrating the nerve cells to create a divergence to multiple pathways. I might have convergence, means I have multiple inputs of information that will allow me to get only one output. I might have convergence in a single source. I might be forming reverberating circuits. And my question here, how do you store information on a flash drive, on a disk drive? What do you think? If you have a memory drive, how do you store the information on this memory drive? If the signals are in the form of electrical impulses that will be transported through wires, how do I store information? How do you store your files on your device? In the form of circuits. So I keep the signal in a circuit like this. And this is how you actually enhance your memory, by reinforcing the circuit. So if you kept reviewing an information that you don't want to forget, what you are actually doing is reinforcing this circuit to strengthen the connection between this set of neurons. And by this way, you would be able to recall the information whenever you need. So when you are studying for an exam, you're memorizing, what you are actually doing, you are creating a circuit between your nerve cells for this signal to be kept in a loop, ready to be retrieved on the day of the exam. Yes, you can consider it an autosave, but theoretically speaking, you should be able to remember everything, but it's not easy to build up those circuits. So you need a lot of input information to reinforce those connections. All right. I need to reinforce, keep reinforcing. And that's why if a memory is associated with a lot of feelings, for example, it will be much easier for you to recall. So, for example, if like, for example, I'm asking you about the day that you broke up with your girlfriend or your boyfriend. I will ask you, do you remember what were you dressing on that day? Yeah, you're gonna remember. What did you eat that day? Although if I did ask you, do you remember the day before that day, do you remember what did you eat and what you were wearing at that time? You don't remember the day before, but you remember this day, this specific day. If I ask you, do you remember the next day after you broke up with your boyfriend or girlfriend, do you remember what did you eat on the next day? You won't remember, but you're going to remember this specific day because it has been associated with a lot of feelings that helped reinforce this memory. The birth of your child. Exactly. You're not selecting, but because it has been reinforced, those connections have been strengthened and will be kept. But because the other memories haven't been reinforced, so you simply gonna forget them. And this is what we're usually asking students when they memorize something, to keep reviewing the information over and over to reinforce those connections between the neurons for you to build a stronger memory that lasts for longer. You don't just study a thing for one time and leave it for a year and expect yourself to remember it, because most of the people won't be able to remember it. All right, so you need reinforcement. That's why also a better way to memorize is to keep repeating the information, read it, write it down. You are exposing yourself to the same information in different means, and this helps you to reinforce the connection between the nerve cells, allowing you to be able to build a stronger memory of the information. All right, moving on to a homeostatic imbalance that is related to the nervous system, and this is the multiple sclerosis. Multiple sclerosis, or MS, it's an autoimmune disease. What I mean by autoimmune disease, it's a disease resulting from a misrecognition of the immune system to my body organs. So my immune system starts to attack my own cells. This is what we consider an autoimmune disease. So what actually is targeted by the immune system? It's the Schwann cells and the myelin sheath. And if I'm attacking the myelin sheath, what do you think is gonna happen? If I'm attacking the myelin sheath and this myelin sheath gets replaced by fibrous tissue like this, that's why we call it multiple sclerosis. I lose the insulation. So I will have shunting and short circuiting of the nerve impulses. I've lost this insulation of the axons. What else? What's going to happen to the speed of the electrical conduction if I've lost the myelin sheath? The electrical conduction speed will be reduced. So I will be slowing down the velocity of the electrical conduction along my axons. And how is this going to be manifested? What parts are going to need very fast electrical conduction? Vision, for example. I will have visual disturbance. I will have weak or slow muscle control. How do you speak? You're breathing air out, and this air vibrates your vocal cords, and then you're going to need to modify those sounds emitted from your vocal cords by contracting, relaxing a large group of muscles, including your lips, your pharynx, your moving your tongue, the muscles in your cheeks. So you're going to be modifying the sounds by sending a lot of signals to the different groups of muscles on here for you to transform the sound emitted from your vocal cords into a language. So this requires a very fast electrical conduction. So try to imagine the number of movements that you are performing by those muscles in order to modify the sound. So for example, to give you an impression about how important are those muscle contractions, can you say "oh" without pursing your lips? What do you think? Can you say "oh" without pursing your lips? You won't. All right, so try to imagine "oh" is just one letter of many that you are using to speak. And this requires very fast modification of the shape of the oral cavity of the lips. So it requires a very high speed of electrical conduction. That's why one of the first things that will be affected is the speech. Will have disturbance in speech. Another thing that will be also affected is going to be the muscles that will be holding the urine in the urinary bladder. So normally micturition, it's going to be taking place once the urinary bladder is filled. This will cause the stimulation of the parasympathetic nervous system, causing the urinary bladder to contract, squeezing out the urine. But in the same time, you need to relax your external urethral sphincter. So the external urethral sphincter, it should be kept closed in order to regulate the passage of the urine out. And in order for it to be kept closed, to be kept contracted, you are continuously sending out signals to this sphincter. So if the electrical conduction, it has been disturbed, so no enough signal is going to be reaching this external urethral sphincter, and as a result, it won't be kept contracted. And if it relaxes, I will lose control of the urine. Person here develops urinary incontinence. Multiple sclerosis, it's a progressive disease that will have a partial remission and exacerbation. Questions? All right, so this completes our discussion of the nervous system today.